bacterium finds itself in a puddle, it's at low cell density. When it finds itself in your gut, there's a trillion cells there, right? It's at high cell density. So commensal bacteria also have to know the difference between I'm out in the environment and I'm inside the host to carry out all the good tasks, right? Because they're not metabolizing vitamins and making, digesting your food when they're out in a puddle. They're living, that's the subsistence farmer life, right? And so I think that, and I do want to make it all be quorum sensing. They see other things like your body temperature says you're in, right, versus the temperature of a puddle. So they're taking in all kinds of chemical and physical information. And I think that when they see those molecules, it means you're in. Turn on the program. Like the only time they're in these big communities is when they're in or on the host. It's not when you're living out on a blade of grass, right? So that's what we think. Mm-hmm.
Is there like a strategy for tricking them into thinking that there's more of them so that they activate and then the body can mount a defense against them?
Yeah, that's a great question. So I will say that one again because answer is yes. So this I think is a fabulous strategy. So the idea is, so quorum sensing's optimized. Right? I mean, if this ends up being true, which at least some of it is true, quorum sensing is optimized, right? They're counting. They recognize when there's a lot of them, and then they do this thing. So what I talked about is you give this anti-quorum sensing so that they don't recognize they're in a group. The analogous strategy is, what if I just add a lot of the molecule right away? So I'm alone, and I turn on my quorum sensing jobs, and I'm making all this stuff, but nobody's there, right? That will also work. So I think if you make the bacteria so that they miss time either too late or too early, show their stuff too early or don't show their stuff at all, I think both of those strategies is going to work. When you go in public and then they make these tapes of you and you talk about making these hyper-virulent bacteria, it's very unpopular, right? So I actually use the other logic, right, for the talks. But of course, we're getting those pro-quorum sensing molecules because I think you just have to, it's optimized, and you just have to screw them up one way or the other, and that will work. In fact, we know that because we have mutants. For example, we have mutants that can't talk, so they think even when they're in a crowd, they think they're alone. They're avirulents. We have mutants that are going, blah, 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 right? They always think they're in a crowd. We could just make mutants, right, that always think they're in a crowd, even when they're not. They're avirulent too. So I do think both of those strategies would work, but that one's a little harder sometimes for the venture capitalists to come to terms with. I don't know why this isn't going anywhere. Yeah, anyway, yeah, it's a good question.
With all due respect to the 20 to 30 cohorts, I just graduated. I'm 51
Congratulations.
years young.
Way to go.
And I'm fascinated with biofilms, and I'm wondering how they're going to be broken down. Pat Gillivet at George Mason is doing some really neat work on Crohn's disease. And I mean, Dr. Reut, my microbiology professor, had a big thing on brushing the teeth. That's really great. But besides the teeth, what other ways of breaking down biofilms are there?
So biofilms, right, are all the rage right now, in a bad way, because in fact, so until about a decade ago, when all of this stuff started to happen, and the realization that bacteria live adhered to surfaces in what we call biofilms, right? It was always thought that they're just floating around making you sick, and that is not true. They are adhered to surfaces, and so now what we realize is when they cover themselves in this goop, this shield, it makes the antibiotics hard to get in, and that's part of why the antibiotics are so ineffective, right? So now, I'm not going to be able to answer your question, right? Because biofilms are the worst problem in the medical field right now. You guys know, you go get a catheter, you get right? Those are all biofilms, and they're notoriously antibiotic resistant for multiple reasons. First, the bacteria resistant, and they also protect themselves in the biofilm, right? And so everybody, besides quorum sensing, so if you don't have quorum sensing, you can't build the biofilm. That's good. But lots of other behaviors like adhesion, like secretion of this exopolysaccharide matrix, those are all the hopefully Achilles heels that everybody's going after now, because it has to be that we treat biofilms. And so there's obvious things. You have to make an adhesant to sit on the surface, right? You have to make the goop, right? So any of those kinds of things. You have to have water channels, to let nutrients go through the goop, right? So there's a lot of behaviors that we know are critical for biofilm formation, and people are trying with analogous screens for their favorite behavior to find molecules that disrupt those processes. And so that's on everybody's radar screen. It's a really good idea, right? But the answer's not there yet, or else we'd be, right? Hi.
I'm just curious how this affects, or how higher animal cells identify friendly bacteria versus unfriendly bacteria, and even, friendly bacteria in places that they shouldn't be.
Exactly, yeah. So this is also, you guys, so, the last 15 years, right, these three questions, this social lives, this biofilm thing, and this idea that these bacteria are these commensals and that they are selected by our immune system to stay and go, right? That there's always this, you get to stay, you get to go, I think are the three most actively studied parts of microbiology today, right? And so we don't know the answer to that. We know that you inherit lots of your antibodies, you inherit bacteria from your mother when you're breastfeeding, right? You get, the reason that you're screwed up like your family members are is because you also inherited the bacteria besides their, and so we know some things about that, right? But how your immune system, they all look like bacteria, like how your immune system says you get to stay, these trillion cells get to stay, and these other few cells that get in have to go, we don't know. But Laura Hooper's doing beautiful work on trying to figure that out. And then it's not just your immune system, it's also these commensal bacteria. So that consortium that you have, your personal microflora, my personal, right, is incredibly robust and actually is playing an active role in keeping invaders out. So it's both your bacteria and your immune system working hand in hand once they pass the test to keep these invaders out, and no one understands how that happens. But it's a hugely studied area now because it's remarkable how it works. And then the same thing you say, you have all these good bacteria, but if they get into the wrong place, they're bad, and then your immune system says, out you go. It's amazing, right? But I'm just reiterating what you said. I don't know how that works. No one knows how that works yet, but it's a big field. Because if we could make that better, right, that would be good. Yeah.
In terms of multiple species working together or existing together and forming a biofilm in an orderly manner, if they all use the same molecule in the second system, have you looked at variables in terms of how fast one species processes the molecule, or at what concentration level?
Right, you guys can come back to Princeton with me. Yeah, so that's right. So, absolutely, right? So one part is we have to know that there is this set of molecules we know we're missing. That's clear. It's still not enough, even if I make more and you make less, right? But we do know that there's all these beautiful engineering feats in these systems. So for example, some of the molecules, the intra-species communication molecule is incredibly hydrophobic. That long tail is so it stays close to the maker. The second molecule with all those oxygens on it is incredibly hydrophilic, so it goes really far. So we're starting to understand that there's a spatial component to the, so it's more than just the molecules building up.